Hello and welcome to the first biology video on this channel. Today we'll be looking at the first sub-chapter in GCSE Biology 9-1. The first chapter is cell structure and transport, and 1.1, the world of the microscope. So today is completely about the microscope. Okay, so the first thing I thought I should go over is not exactly biology, but it's very important. You should already know it, but just in case you don't exactly, or you're not very confident with it, we'll go over it. And that is the scale, the scale of numbers, the scale of units. So let's take a very common example here, which we'll be using for microscopes, So the common meter, meters, kilometers, centimeters, you should know all the conversions at your fingertips, and you should know what these prefixes actually mean. Not this one, obviously, because this is an SI unit by itself, but centi, kilo, milli, micro, nano, as such, for all different units, not just meters. These prefixes you should be able to interpret in all circumstances, not only in biology, but in chemistry, physics, math as well. Okay, so let's look at what this exactly means. So we have a meter, which is the normal SI unit, right? It is the SI unit. It's the standard unit. Okay, so the SI unit. And how do you get from kilometers to meters, right? You would multiply by 1,000. And what does that mean? If you multiply by 1,000 to get from kilometers to meters, that means that in a kilometer you have 1,000 meters, right? This is common knowledge. Then from meters to centimeters, again, times by 100. Actually, the normal one in science, usually you'd go from meters to millimeters immediately, because this should make sense. If we go from centimeters to millimeters, it's times 10, right? So times 100 times 10. So centi is just something in the middle we have because it makes us easier. It makes it easier to measure daily life, things in daily life. So anyway, from meters to centimeters times by 100, what does that mean? We have 100 centimeters in a meter. And then if we go from centimeters to millimeters, what does that mean? We have 10 millimeters in a centimeter. And then from millimeters to micrometers, we have 1,000 micrometers in a millimeter. So you would multiply your millimeter value by 1,000 to get the value in micrometers. And then so on for nanometers, which is as small as it'll get, at least in this microscopes topic, right? So times by 1,000, you would get to nanometers. That's how it works. As well as that, obviously, you should kind of keep it in your mind. You don't want to be listing this out in questions. You won't. After you get a lot of practice, you'll be able to do it like in your mind. It's not that difficult, honestly. So if you wanted to get from nanometers to centimeters, for example, what would you do? You would do the opposite, right? You would divide by 1,000, divide by 1,000, divide by 10. Right? You can do this in order if you want, or you can just do it together in one simple calculation. Just like the meters, centimeters, I think I told you here. You want to get from millimeters to meters. Well, here you are multiplying by 100, then by 10, so that's multiplying by 1,000 in total. Sorry if this is messy, but yeah. Then you get from millimeters to meters. You do the opposite, so you divide by 1,000. Actually, you divide by 10 and then divide by 100, but in one simple calculation, it's divide by 10 times 100, divide by 1,000. It is actually a very, very simple concept. You should get the hang of it as soon as you get some practice. Okay, so on to the next part, I would say the most important part, which is what microscopes actually do, right? So microscopes allow us to view specimens. Specimens are just basically things we observe, okay? Things we observe. So view specimens that we cannot observe with the naked eye. So things we can see with the naked eye, I don't know, a pen, the Apple Pencil I'm holding in my hand, anything basically in small objects. We can sometimes see insects, right? Very small organisms, but we can still see them. But we have microorganisms like bacteria and viruses that we definitely cannot see with our naked eye. So there are two types of microscopes that help you see things like this. And let's make use of a table to understand this, probably the best way. So we have something called a light microscope and something called an electron microscope. The electron microscope is very sophisticated. It's used in very big organizations, but in a school or in a simple laboratory, you would just use a light microscope, and that's the common microscope you have in your head as an image probably. Okay, so first, operation of both of these microscopes. A light microscope simply uses a beam of light and lenses to produce an image of the specimen. That's what it does. You just need to know that. However, an electron microscope, self-explanatory, uses a beam of electrons to produce an image of the specimen. It's in the name, electron, a beam of electrons, a beam of light to produce the image, okay? And then your max magnification, the maximum magnification. What is magnification? I think you've definitely heard of it before, but what we mean by magnification, if you do not know, is how many times bigger the image created by the microscope is compared to the actual size of or whatever you're looking at, the specimen, right? So if I were to look at a bacterium, right, a bacterial cell under a microscope, let's say this light microscope is giving you times 2000 magnification. That means that it's going to look 2000 times bigger than it actually is in real life. Okay, so the maximum magnification that you would find in the best light microscopes, times 2000. However, in an electron microscope, which is much better, you would find a magnification in some cases of times 2 million. Okay, so that's a huge difference. Next part is the price. In terms of price, the light microscope is definitely cheaper, which is why you find it in schools, simple laboratories. However, an electron microscope you would find in sophisticated laboratories, right? It's much more expensive, much more expensive. It's huge. We'll come on to that. So anyway, then conditions, right? Conditions. A light microscope can be used anywhere. There are no specific conditions. You can use it at home. You can use it in a lab. You can use it in the school. It's fine. It all works. It's all good. However, an electron microscope is large. As I said, it's very big, much bigger than a light microscope. It must be kept in rooms where temperature, humidity, and pressure are carefully controlled. So basically, you need very specific conditions for the electron microscope to be working correctly in the way that you want it, right? To get some good results. And resolution. What does resolution mean? Well, the textbook definition of resolution is just, the shortest distance between two distinguishable points. That can seem confusing. So it's much better to just express what we mean by resolution using images. So this image of a microscope here has a pretty good resolution. An image of a microscope with a bad resolution is this. See, look at that. This is kind of blurred, while this is a very good resolution. Okay, so that's what we mean by resolution. And that means this one thing you need to keep in mind is the smaller the number for resolution, right? The better it is. Because as I said, what we mean by resolution again is the distance between two distinguishable points. The distance between two different points that we can make out. If I blurred this out as much as possible, I would just see a white space, right? So the distance between 2 points, as I would think, is like infinite, right? Because I can't find two different points. However, if it's not blurred and it's really clear, it's a very small distance between two distinguishable points, which is why a smaller number, remember this is distance, this is nanometers. So if it's very small, that's much better. So as you can see, the electron microscope has a much a better resolution, and this is a lower resolution despite the number. You need to keep that in mind. It could get confusing. I know it says here 10 nanometers or 0.2 nanometers. That's because there are two types of electron microscopes. We're going to come on to that in the next slide. Nothing that you need to learn too much. It's just a small thing you need to keep in mind. So specimen type. What type of specimen can they see or can they show us, right? Create an image of. So a light microscope can show you everything. Okay, this is one of the only advantages of a light microscope, apart from the fact that it's cheaper. And it can be used anywhere. Okay, so all types of specimens. It can show you anything, including live ones. However, an electron microscope cannot show you live specimens, as in the specimen cannot be alive. The reason being the beam of electrons would kill your specimen immediately if it is alive. Okay, so it cannot show you a specimen while it is alive. It can only show you dead objects. It's still useful. Many things like DNA or even bacterial cells, even if they're dead, they're very useful because they still retain their intracellular structures. We'll come on to that. An electron microscope is very good. It's just that it's very expensive, needs specific conditions, and it can't show you live specimens, which are useful in some cases. So now coming on to the two types of electron microscopes. We have one called the transmission electron microscope, and then we have the scanning electron microscope. These are the only differences you need to know between them. It's just that the magnification in a transmission electron microscope is higher. A scanning electron microscope has a lower magnification. You just need to memorize these things, just know them at the back of your hand, just in case they ask it. It is in the textbook, it is in the specification. So the resolution as well, transmission electron microscope has a better resolution. A scanning electron microscope has a lower resolution, right? Because remember, the greater the number, the lower the resolution. Image type, a transmission electron microscope shows you 2D images, while a scanning electron microscope shows you 3D images, however, at a lower magnification, right? A lower magnification and a lower resolution, but it can show you 3D objects, right? Okay, so the next part is a very key part, and that is a golden equation that you need to know, because many math questions in biology, it's not much of math. I think around 10% of the questions in your paper will be math, compared to chemistry, which is 20%, physics, 30% of the questions you're asked are math, approximately. But still, it is quite important in biology as well. So magnification, we saw what magnification was. It's a number, remember, like times 100, times 2000, for example. Yeah. So magnification is equal to the image size over the real size. What do we mean by that? The image size, so literally the size of the image, okay? The size of the image. So let's say I have a cell shown over here. We'll look at cells in the next topic, but just how can I say? It's a unit of an organism, okay, or a bacterial cell, okay? And we have the real size of it. So let's say it's like this, right? By image size, I just mean any particular distance that you can measure on both image and real. So for example, you could measure the distance between this end and the distance between this end. That is your image size. And then you could measure the distance between this end and this end. So the same 2 points, but on the smaller image, or on the real size. So this. So if you have this distance, x, and you have this distance, y, your magnification would be the image size, so that's y, by x, divided by x, right? And that would give you the magnification. And doesn't mean you can do it from anywhere, okay? Anywhere on the cell or anywhere on the image, as long as it's the same for both. So I don't need to do this. I could have done, this instead. And then with the smaller real image, this as well. If that makes sense, try to understand that. You could do it from anywhere as long as they're the same in both comparisons. It's the best way I can explain that one. Now let's see how we use this equation in an exam question. Okay, so first, the best way to approach any exam question is to read it carefully. I know that sounds funny, because why would you not read it carefully? But trust me, so many students, they read a number wrong, especially in numerical questions, they read the number wrong, or they don't follow every instruction. I've done that as well. I'm no exception. I've always forgotten the last part, especially relating to your units or your, the number of decimal places you have to round it to. But anyway, an onion cell is 6.2 millimeters wide. So remember that in your mind, that it is millimeters. I'm just going to write that again. Millimeters wide when viewed with a magnification of times 200. The minute you read that magnification word, you should be thinking, okay, I'm going to need to use this magnification equation in this question. And then find the real width. Okay, the real. So now we need to find real. We have the image because it's viewed under a microscope with a magnification. So your image size is going to be 6.2 millimeters wide. Keep that in mind. We need to find the real width. And they're following the instructions as well, because width, remember they're measuring the same thing of the cell in centimeters. Remember here we have millimeters. We need to find it in centimeters. Keep that in mind. Finally, express your answer in standard form. You should already know how to be using standard form from math or from your teachers for a long time now. If you don't, I really recommend that you go and refresh your skills on that, okay? So now let's actually do this question. So let's derive the equation first. We know the equation, right? Magnification is equal to image size. I'm just going to say image width in this case, because the size can be anything you want. Image by real. Now we know the magnification 200. I'm not going to say times 200 because the times is just a sign. 200 is equal to the image, which is 6.2 millimeters. Okay, 6.2 millimeters over the real. Okay, over the real, or R, if you want to call it R, whatever it is. Okay, so 200 is equal to 6.2 divided by the real, which is R. So now you can just rearrange the equation. So that means bring R to this side and bring 200 down there. You should know this from math, very simple math. If not, please go over it. Okay, so that means R would be equal to 6.2 divided by 200, right? What does that give us? Well, if you divided a 6 point, you can use a calculator, by the way, in biology, in all three sciences. That's completely allowed. But what you can do here, obviously, is just divide by 2. What I would do in my brain, divide by 2 and then divide by 100. So you would say 3.1, and then you would divide that by 100. So what does that give you? 0.031, right? Because 6.2 divided by 200 is 0.031. But what is that in? It is in millimeters, because we use millimeters here and we're having magnification. So this would also be in millimeters, but we need to find it in centimeters, they're saying. So how do we convert from millimeters to centimeters? Well, we divide by 10, don't we? Because remember I saw in the scale, we would divide by 10. So if we divide by 10, that is equal to 0.0031. You just move the decimal point to the left, right? 0.0031 centimeters. That is our answer. However, express your answer in standard form. I've gone all in designing this question, putting as many tricks up there as I could, as many steps as I could. So 0.031, that you should know immediately. Standard form is 3.1 multiplied by 10 to the power of minus 3. Right? Because it's minus 3 because you're moving the decimal point 3 times, from 3.11 to 3. And the number that you're expressing in standard form has to be between 0 and 10, 1 and 10. So 3.1 times by 10 to the power of minus 3 centimeters is your answer. That is the width of the real cell, of the onion cell. So now, let's take a look at the microscope itself and the steps you would go through to view something. Okay, so first of all, quickly going over the microscope, you probably won't get asked to label your microscope, but there's no harm in going over it. You just might. They completely have the authority to ask you about it. So this is something called the eyepiece, okay? Straightforward, it's what you look through. Usually has a fixed magnification that you can't change of times 10 already. These are called the objective lens, okay? And as you can see, there are 3 here. You can just rotate them around. You can rotate them around and put what magnification you want. Okay, they have different magnifications. Now you have the coarse focus knob, okay, and the fine focus knob. Okay, well, what does that mean? Well, the coarse focus knob allows you to adjust the stage up and down. Okay, it makes the stage go up and down as you rotate it until your object comes into rough focus, until you can see your object. And then fine focus, you rotate it until you can actually see it very clearly, the specimen very clearly. The slide is something you place on the stage. You take your slide out first, and then you place something on it, whatever, it's a cluster of cells, a plant cutting, and then put it back on the stage, and then view it with your eyepiece and your objective lens. Okay, of course you have the light, which allows you to see it because the light is projected through the stage and the slide, and you can see it properly. That's how a microscope works. Now for the steps, if you were asked a 6-mark question in an exam, for example, how would you view an onion cell? How would you view this plant cell? Right? This is how you would view it. So this first step, add a drop of water using a pipette to the center of the slide. So you remove the slide first, but that doesn't need to be a step. You can include it if you want. To the center of the slide. Then you place your specimen into the water on the slide using tweezers. Okay? On the slide using tweezers. Okay? So the specimen could be, cutting of plant or cutting of a leaf, right? A small part of a leaf. It needs to be extremely small so it can fit on the slide, and you can see it through the eyepiece. Then you add a drop of iodine solution to the specimen. Iodine solution, okay, it's a blue-black kind of, it's an orange-red kind of solution. What does that do? When you add it, it stains the specimen so it has a color, so you can see it more vividly, okay? And then you have, basically you're placing a cover slip onto the slide. That's just a transparent, let me write this here. It's just a transparent slip that you put over. So just to protect and cover the specimen. You would fix your slide now onto the stage like it's shown here. See, it's clipped on with some tape, but sometimes you would see tape, sometimes it actually has the clips for you to put on. Rotate and equip the objective lens. Okay, so these are the objective lens with the lowest magnification. So each of these three have different. Each of these three has a different magnification. You would equip the one with the lowest magnification. Then you would look into the eyepiece and rotate the coarse focus knob, this one, until so that would move the slide up and down, right? Until the specimen is roughly visible. Okay, first you would see nothing, presumably, usually you would see nothing, you would just see a blank white. And then as you rotate this up and down, play around with it a bit, you would kind of see something. You kind of see that there's something there, there's some kind of specimen there. Then you stop. Then you rotate this focus knob, this one, the small one, fine focus. So, sorry, this should be fine focus. The fine focus knob, until the specimen is clearly visible. Until you can see it perfectly with perfect resolution, until it's not blurred. Because here, it would usually be blurred after this step. It would be in a low resolution. Okay, this one makes it the highest possible resolution that you can find. And once you're done with that, you observe your specimen and do whatever you need to do. And that's it for chapter 1.1 in biology. I'll see you in the next video.